Now we will see how we can find out the conserved patterns within the DNA sequences. So as already we know that there are two possibilities: either we do not know the location and the size of the pattern, and second is there is a possibility and chances of mutation within these patterns. So to overcome these problems, we will rely on the alignment matrices. So now we will see how these alignment matrices can be worked out. In this slide we can see the sequences of a very specialized pattern of NFKB binding sites of the Drosophila melanogaster. So here we can see the 18 sequences from 1 to 18, and the conserved pattern sequence is this one, but we can see due to mutations this pattern is not present conservedly in any of the 18 sequences. So this problem can be overcome by aligning these sequences, and we can count the number of A's and T's and C's within a column J. For example, in the first column, number 1, we can see the most abundant nucleotide is thiamine and its number is 12. And if we see the next column, in this column we can clearly see that the most abundant nucleotide is cytosine, that is 12. There are also other nucleotides, but their number is less than 12. Guanine is 0, thymine is 3, and adenine is also 3. So in this way, in all the coming columns, we will calculate the number of nucleotides: adenine, thymine, guanine, and cytosine. Nucleotide that has the maximum numbers will be counted as and circle and written in the bold red letter. So in this way all the nucleotides are counted and it's bold in red. For example, in this column adenine is maximum 16, in the next column thymine is maximum 9, and the last column again cytosine is maximum 11. So the maximum number of each column is highlighted in red, and after calculating the highest number in each column, we can now draw the consensus sequence of this motif. So the consensus sequence will be, in the column number 1, the maximum number of nucleotide was thymine and its number was 12. So bottom of the first column we will write thymine. So in the second column we can see the maximum number of nucleotide were cytosine. So we will write C here. So in the third was G, in the fourth again G, and so on. So the maximum number of nucleotides present in the column will be written in the bottom of the column. So after writing these nucleotides, we will get the consensus sequence of this NFKB binding sites of Drosophila. And similarly, we can also calculate the score of this matrix. So we can see here in the first column it is 12, and we have written is 12. In the second is also 12, third is 16, then 18, then 14, again 16, and so on. And then we will count all these numbers and it comes out to be 159. So this is the score S of this profile. So we can count the score S of any of the DNA sequences that are aligned in this manner, and we can, by using the technique of alignment matrix, we can calculate the score of these matrices. So in this figure number 6, we can see here, because we have the sequences of DNA, and we remember in the previous slides we have incorporated a pattern P within the 7 sequences of the 7 DNA sequences. So we have incorporated, and we have noticed that there are 2 mutations within each pattern. So, and we have also noticed that, for example, if we want to find the sequence of a motif, for example, this is gene number 1, this is gene number 2, 3, and so on. And this is the promoter region upstream of the gene. So, for example, there is a conserved region within this promoter region, and in gene number 1, this is the location of this motif. In gene number 2, this is the location, and gene number 3, this is the location. So we can see the location of all the 3 motifs are different, but the sequence of these motifs are almost conserved, if not very much different. So if we want to find the sequence of all these 3 or more than 3 motifs, we can use this technique of alignment. So we should know that in this case, the sequence number 1, this one, we know that the starting point of the conserved pattern is nucleotide number 8. Here is the position. In the second sequence, we know that the starting position of this conserved motif is 19. So it is 19. Similarly, in the third sequence, the starting position of this conserved motif is 1, 2, and 3. And similarly, the positions of this conserved pattern is written within all the 7 DNA sequences. As we know that this sequences, the starting position is very different. So to overcome this problem, we have to align these sequences. For example, here in this case, the position of all these motifs are different, but we have to align this, we have to move these sequences so that in this case we can see here all the first conserved motif will come in this position, whether it is starting from 8 or 13 or 3, and we can see this motif is come in this area. So we have to shift these sequences so that in this case also we have to move this so that all of these will come in this area. So we have to shift this gene 2 here and G3 in this direction so that they will come in the alignment. So we have seen that we can calculate the consensus sequence of any of the conserved region within the promoter or any other region within the DNA by using the technique of alignment matrices.